Okay, welcome everybody to today's webinar. If anybody's having any technical difficulties, they can call 518-402-8028. And at this time, I'll turn it over to Kathy. Kathy?
Thanks, Greg. Welcome, everyone, to the session number 2 for the 2026 Winter Webinar Series for the Northeast Aquatic Plant Management Society. And okay, slides are not working currently. I don't know why. They were working before. So I'll just hold on a moment. Sorry. Let's try again. There we go. Okay. So I'll give you a little bit of background information for the Northeast Aquatic Plant Management Society. Our mission is to assist in the management of aquatic vegetation, to provide for the scientific and educational advancement of members, to encourage scientific research in all facets of aquatic plant management, to promote an exchange of information among members, and to extend and develop understanding in the discipline. Our members are from academia, government agencies, private consulting firms, herbicide manufacturers, and lake associations. We have annual meetings, annual plant camps, webinars with CEUs. Unfortunately, that's not the case for this particular webinar series, this session. And we do offer student scholarships up to $10,000. Encourage you to visit www.neapms.org to learn more about the society. All right, sorry, session 2. I forgot to change that, but the rules are the same. So this is session 2, webinar set to run from 1 to 3. There'll be 2 presentations followed by Q&A at the end. Please type your questions into the chat box and the moderator will read them during the Q&A session. Please note we are not able to offer CEUs for this webinar. The webinar will be recorded and will be placed on the NEAPMS website in the future for viewing for other folks who've missed or would like to review the content. Today's presentation to speakers are from 1 to 2, ecosystem risk and mammalian susceptibility of the stable lipid-soluble toxin Aetokthonotoxin or AETX from the epiphytic cyanobacterium Aetokthonos hydrillicola. Speakers, Dr. Susan Wild from Warnell School of Forestry and Natural Resources at University of Georgia in Athens. From 2 to 2:30, we have RT-PCR assay development of ITS and toxin gene regions of Aetokthonos hydrillicola, a known epiphyte of the invasive species Hydrilla verticillata, and that's by Dr. Teresa Hattenrath, the New York State's Department of Health, Wadsworth Environmental Biology Lab. Whoops. So we will start without further ado. Introduce Dr. Susan Wild. So Susan is professor at the University of Georgia's Warnell School of Forestry and Natural Resources. Her research and teaching focus on impaired aquatic systems and challenges with invasive plants and animals, nutrient pollution, harmful algal blooms, emerging contaminants, and their implications to fish, wildlife, and even humans that depend on these valuable water resources. Dr. Wild discovered the cyanobacteria Aetokthonos hydrillicola, which produces the neurotoxin responsible for killing bald eagles and other aquatic wildlife in reservoirs across the southeastern United States. Dr. Wild received her PhD from the School of Forest Resources at the University of Georgia. Please take it away, Susan. I'll stop sharing.
Well, thank you for inviting me, Kathy. You've given me an opportunity to tell this story to a lot more people. And I'm going to start way back 25 years ago to tell you how this research started today. But yes, it's about hydrilla and a toxic cyanobacteria that grows on hydrilla. And lots of things eat both of those in that food chain. So we know that cyanobacteria can produce nasty toxins, dangerous toxins for animal and human health, as well as for fish. And we've seen a lot of planktonic water blooms. You're probably most familiar with that. Looks like paint scum on the water almost when it gets really thick and can cause water-soluble toxins that are bad for people. I've been working on epiphytic algae growing directly on the leaves of aquatic plants, and we're seeing that this edge environment is very critical. This story will open up other plant and algae research connections, I'm sure, because we haven't been looking at the algae attached to the plants and attached to the sediments as much. We've been more focused in this water column, which is very critical because that's our water supply. But in the benthos, dogs are getting into some of our rivers and dying from interacting with that benthos. So we're trying to keep people mindful of the fact that it's not just in the water, but on the edges of things as well. So blue-green algae or cyanobacteria are kind of between the bacteria and algae. They can photosynthesize, but they're prokaryotes. They can fix nitrogen, so they live in really extreme environments. And they have some very specialized pigments, which show up even from space. You can see if this pond has a dangerous blue-green algal bloom, a cyanobacterial bloom in it. And the farmer who had us come out there lost 5 of his prize cattle drinking out of this pond before we convinced him to water them off site, as this was not safe to drink out of. So he didn't realize that just looking at the color of this pond, he should have probably had some trepidation about letting his animals drink from it. And I know a lot of you probably wouldn't let your dog swim in a pond that looked like this. Microcystis aeruginosa is the most common bloom forming in the water column. And in fact, that was the picture I was showing you. That was a microcystis bloom. And Microcystis aeruginosa produces a liver toxin almost all the time when it's present. And it's microcystin, and that's quite a dangerous liver toxin. It can cause, well, initially they called it the fast death factor before they named it microcystin. Because of the injury to the liver and spleen and kidneys. It can even be genotoxic, cause cancer to the lungs and intestine. We have had blooms like this in Lake Erie that have affected the drinking water in the US as well. This is our campus swimming lake, not swimming anymore, but they do let them paddle around in it. This particular bloom was not producing toxins at the time when I sampled it, but you frequently get these kind of water column blooms in the fall. So that was our campus representative. But the ones we worry about the most are the neurotoxins, and these can be produced in the water column. And there's some examples here. Anabina, oscillatoria, and Aphanizomenon all can produce these water column blooms that have neurotoxins, which were originally called very fast death factor. Anatoxin A, saxitoxin, and these cause immediate death of livestock, dogs, and birds. So as I said, I'm going to focus on the epiphytic algae today. This is a new species that we discovered when eagles were dying in Arkansas long ago. 1994 is when it first occurred, and they called it the eagle and coot disease because they didn't understand why all of these eagles and coots were dying with these lesions in their brains. So they named it eventually, when it was described, avian, bird, vacuolar, holes, myelinopathy, a disease of the brain, pathology of the myelin. So I've been working on this for a while, me only since 2001, but this first presented in the scientific community in 1994, and a lot of reservoir monitoring was done. We started doing field studies and food chain transfer trials to look at the expanding locations where this was occurring, and we were able to eventually determine that the cyanobacteria was producing the toxin and fully describe that toxin, as well as the gene cluster to produce the toxin. So unfortunately, I'm at the end of the rope trying to tell people that yes, this new toxin kills eagles, but it would also be a risk to human health if we consume fish and waterfowl that have been exposed. So I'll back up and start at the beginning, I promised. In Gray Lake, Arkansas, in 1994, apparently 2 fishermen were out on a boat and found 2 dead bald eagles. And they turned it in to the authorities, and they started monitoring for them at DeGray Lake and found 29 more dead bald eagles that year. So they pulled together a group of researchers, especially the eagle experts and toxicologists, to try to understand why we had this mortality event in an isolated reservoir in Arkansas. They didn't they were studying it the following year and didn't see any wildlife deaths. So they're like, well, maybe it was a one-off. We're okay. But then the following year, once they disbanded their research efforts, 26 more eagles died, and it was in additional reservoirs throughout the state. So we, at that point, understood that this was the most significant unknown cause of eagle mortality in the history of the U.S. And this is a picture that Tom Murphy took. He's an eagle biologist from South Carolina. There's an eagle overflying a raft of coots. They try to bunch up together to look like one big bird, I guess. But in any case, they are afraid of eagles. This is a little video of how sad it is when the birds have consumed the hydrilla with the neurotoxin on it. The eagles have drooping wings. The coots, when they come up from diving down, will end up upside down in the water and can't seem to right themselves. When they're on land, the coots just look drunk. They are not able to keep themselves upright. So this is a disease that kills most of the animals that become exposed to it. But there were no consistent findings from the early studies that were done out in Arkansas. There were no infectious disease agent, and there were no known toxins that could cause this type of intramyelinatic edema or open spaces in the myelin sheath. So the brain lesions were the only consistent finding, and that was a very novel thing. We've been looking at eagle brains for quite a long time, since the onset of DDT concerns, and this was a novel finding to see these lesions. So the Southeastern Cooperative Wildlife Disease Study here at the University of Georgia has done a lot of the research into the histology and the consistent findings of this outer myelin sheath that has this holes in it. And once it begins to get so vacuolated, then nerve connections are no longer very effective. So we start to see neuropathy in the animals that are affected by this. This is what it should look like, a nice pink outer myelin sheath. And no, it's riddled with holes. And we knew that the bald eagles, the juvenile shown here, and adults were affected. We found great horned owls with the same lesions who are also nesting on the same reservoirs as these eagles. And the herbivorous waterfowl were also dying at these locations and presenting with the same type of lesions. So as the research progressed, we realized it was more than an eagle killer, and that in fact we should be looking down into the food chain, because these herbivorous birds are their favorite food and quite abundant in the fall when the hydrilla is dying back, and we knew that the cyanotoxin was being produced by our cyanobacteria. So at first we had it in Arkansas, and then Dr. Fisher and crew over at SCWDS, the Southeastern Cooperative Wildlife Disease Study that I mentioned, did a more expansive survey across the southeastern United States and found birds presenting with the same symptoms and the same lesions all the way from Texas to North Carolina. So we began investigating those locations. Oh, I'm sorry. Let me go ahead and finish up what research that Dr. Fisher completed. We knew by the time we started our work that there was the possibility that the eagles were consuming coots, and that whatever the toxin was in those coots, because they collected coots from affected reservoir and fed them to some non-releasable red-tailed hawks, and were able to demonstrate that the lesions appeared in that laboratory hawk that received the coot tissues from a neurotoxic coot. So we started looking at all the reservoirs that were affected. What is the common denominator across this landscape that could explain why we have the hydrilla with the new cyanobacteria? Perhaps we were suspecting cyanobacteria from the very beginning because, well, that's what we do, and they're very dangerous neurotoxin producers. So that was what we were looking for, but we didn't find it in the water. We didn't find very abundant algae. It wasn't in the sediment, but the plants did have abundant epiphytes on them. And so we started focusing on the plants right away and could see that there was extensive coverage by a novel species of cyanobacteria. So we had to describe that first before we could get to describing the toxin. But we got a lot of new locations based on just new hydrilla infestations that we tracked, and then we're able to conclude that it was harboring the toxic cyanobacteria. So most of our sites are hydrilla, but there are sites that have Merophyllum spiccatum and some that have Egeria densa. So, you know, about anywhere in the US, this potentially could be, but I'll show you a model at the end that shows it's not just dependent on where the invasive plants grow, but whether that environment is good for the cyanobacteria to grow on it in that location. So we have some examples here of how densely, this is all the same species of cyanobacteria growing on the hydrilla at Thurman. So this is Aetokthonos hydrillicola on hydrilla from Lake Thurman, and Egeria had the same species. We can find it on these native plants, but it's going to be a lot less abundant on those plants. And I have a couple of reasons for that, but probably mostly because these two guys can hyperaccumulate contaminants, and one of them is bromide, which turns out to be what's in my toxin. So that was a really big boost if the hydrilla or Egeria die back and provide that bromide to my cyanobacteria, it can produce the toxin. Whereas these may not, they don't tend to accumulate bromide, and they don't get very abundant, and the cyanobacteria is not that abundant on them. So I really focus on the invasive species aspect of this because, like any dose response, I don't know if you're familiar with that, but if you have enough of something, it can be very bad for you, even Tylenol. But in a very low dose, it might even be beneficial or wouldn't matter. So I'm talking about dosage here, how much is there. And we continue to track the AVM sites across the southeastern US as we found more new sites. By 2009, we had 17 different man-made lakes. These are all impoundments that we're finding it in. And they overlap in the locations where we'd found problems previously. It looked like it was kind of in the Piedmont across the southeast at that point. And I've had so many wonderful students work on this. This PowerPoint includes a few more of the folks that really did all the hard work out there in the field. But I want to show you a few pictures that just show you what the hydrilla looks like when it has the Aetokthonos hydrillicola on it. These black spots, dark spots on the underside of the leaf are large colonies of Aetokthonos. So you can see it with the naked eye, but we always have to confirm it, not only with microscopy, but with fluorescence microscopy, because under regular light, this just looks like green hydrilla leaf, right? But there's some kind of dark greenish stuff here. Well, if you turn on a fluorescent light on the microscope with special filter sets, you can turn off the chlorophyll A that's in the leaf, and you're left with the cyanobacterial pigments. These are the pigments that only the cyanobacteria can make, so they show up under this special illumination. And Teresa will also show you some pictures with the fancy red cyanobacteria. So we conducted food chain transfer trials to say, okay, it looks like the herbivorous waterfowl are going to be transferring up to the birds of prey, but we need to see where the herbivorous waterfowl are getting it. So we used some game-raised mallards in an experiment where we gave them hydrilla without this novel cyanobacteria and then hydrilla with Aetokthonos hydrillicola. And the only ones that developed lesions were those that were fed the hydrilla with the novel cyanobacteria. So at this point, we knew that this very abundant cyanobacteria that was growing on the hydrilla was dangerous to the mallards in the trial and causing the lesions. And those aquatic plants and epiphytic algae are primary food source for coots and other herbivorous waterfowl that migrate down into our reservoirs in the fall at precisely the same time as hydrilla dies back and makes that bromide available to produce a toxin. So it was like a perfect storm of arriving just when things were getting bad. And the eagles are nesting in our reservoirs. They set up their territories in August, and then they're tied to those territories. They lay their eggs in the spring and they fledge in March and April, but they don't on AVM sites. They just end up dead in their nests. And so it's a pretty clear sign when this eagles, our sentinels, are dying in these reservoirs. We have had other ways to test the toxin because we're all a bunch of bunny huggers or tree huggers here in my lab. So we didn't really want to keep having to kill animals to test the toxin. Using a cell line, you're able to get an idea of what the effects of the toxin are in some bioassays. So that did help us to get an idea of what if we did a hydrilla extraction where we just take out all the toxic components of it, not the hydrilla itself, but the extract from it, then we were able to show that if you extracted the toxin and then use that on the birds, that only the mallards that received the positive extracts from the hydrilla developed lesions. And really the most positive response was in methanol, which is both a little bit lipid soluble, just slightly water soluble solvent. So this gave us a good clue as to the fact that this is not an infectious disease. We can induce this with the extract alone. And was very efficiently extracted with methanol. So we suspected could be a polar compound. And we tested to see if it was heat sensitive. We've been trying for a while to work out in what way could you process it without breaking it down. But the scary part is we could heat it up, we could freeze it. It is actually quite persistent. We conducted our own sentinel trial with mallards in Davis Pond. It's real close to Lake Thurman, which is a giant Army Corps reservoir. This is a small farm pond. And we built an exclosure in this farm pond after they got hydrilla. And we put some mallards into this lake. They were wing clipped, and with the help of a bird dog, we were able to recover those birds, but they all developed AVM by the end of the trial. So we were able to show that in a new location that the sentinel birds were telling us that hydrilla did have the toxin in it. We wanted to figure out what can we do about this pretty early on. So Becca Haney, as a grad student and then postdoc in my lab, worked on this triploid sterile grass carp trial where we, in the same pond, release sterile grass carp, and they will eat all of that hydrilla out of that pond. And then Becca ground up those fish and fed that to chickens, and it didn't induce the lesions in the birds. So we felt like there must not be enough concentrated in their tissues to be able to move it to the next trophic level, but we did find vacuolar lesions in the fish. So we still have some concerns about this as a biocontrol, but it is probably the only way to truly eliminate hydrilla from a location where the Aetokthonos is present. And it does appear that it can work. Once they've eliminated the hydrilla, then the source, the production of that biotoxin will stop if you can interrupt the hydrilla in that lake or pond. So dumping them in there full of hydrilla, then we let them consume all of the hydrilla, and they did. So at the end we were able to show that while the grass carp developed the lesions, they did not transfer the toxin in that trial. And she certainly went through all the steps to try to feed them. And that was published in the Journal of Aquatic Animal Health. And I believe the census is recorded that people will have time to write down references if they want to find those original articles. This was from another sentinel study that we did in Henry County, Georgia, to try using a reservoir that was partially treated. So we looked at some mallard survival in a cove without the hydrilla and Aetokthonos, and this mallard was from the location with and he has tremors and then a seizure. And this bird, of course, had to be euthanized, but it is a scary thing when you see them. So we have this harmful cyanobacteria growing on invasive plants and all these sites, and we were finally able to describe it. And since it was both a new genus and new species, we had to come up with the full name: eagle killer living on hydrilla, Aetokthonos hydrillicola. And this is expanding a toxin food web. Everything we have done laboratory feeding trials with we have seen as vulnerable to this toxin. We recently did a mouse trial that I'll give you more information about at the end, and saw that mammals are indeed susceptible to this toxin. We found beavers and now panthers and bobcats in the wild with the toxin, as well as turtles and eagles. And we have been working with some of the waterfowl and found the toxin in them as well as the fish. But these are from trials that were conducted in our laboratories here at Warnell with multiple professors, including John Mayers and Sonia Hernandez. And this little video of the turtle isn't showing up, but he is dragging his hind leg. So one of the things that we saw with the turtles is they had that hind limb ataxia, which has been described in some of the panthers as well. Sorry that didn't show up. But you could see the same lesions in the brain slides of the turtles as well. And the tadpoles that were eating the hydrilla directly all experienced mortality if it had the cyanobacterial toxin associated with it. So we had control hydrilla, they were doing fine in those, but they were dying off in our treatment exposures. So this is the bullfrog and the leopard frogs. And the one that was probably the saddest to see is the salamanders, because they had almost immediately started to have effects from it. And we had to sort of gut load our prey items so that the paedomorphic salamanders would consume them directly. Paedomorphic salamanders have a longer life cycle. They live in the water for a while until they get to be probably 2 years. And then depending on conditions, they'll become terrestrial and lose all their gills and so forth. But during this stage, they're quite vulnerable because they're eating aquatic prey, and they ate tadpoles and snails that had consumed the hydrilla with the cyanobacteria, and that produced vacuolar lesions and this clinical behavior in the salamanders. The snakes were tougher. They didn't seem to have a lot of behavioral changes other than the fact that they kind of stopped eating in a couple of our treatment snakes, but we did see vacuolar myelopathy in the spinal cord of the snakes, which is interesting, as probably really important for a snake, their movement. And so depending on which organism it is, we may see that there's a different point where it affects the brain tissue, the nervous tissue. But one place we've been working more and more recently is in Florida, because much like the coots, these invasive apple snails can consume a tremendous amount of hydrilla. And then everything eats these little invasive apple snails. Well, actually, they're not that little. They're the size of an apple. The native apple snail, Palidosa, is this tiny one, and it doesn't reproduce very rapidly and only has 100 eggs each time, twice a year maybe, whereas maculata has 1,000 eggs every 15 days or so, like much more productive. So they've overtaken the native, and these invasive apple snails are now being consumed by snail kites. And our concern was that that would work its way into the snail kites as well. So we did an experiment with chickens because snail kites are endangered, so we're not doing experiments on them. But as our bird substitute, we did discover that if we fed those chickens, the snails that had been consuming hydrilla with the Aetokthonos, they did develop lesions. So proof of concept is that if a bird consumes these invasive apple snails that have consumed hydrilla with this cyanobacteria on it, they're going to get sick as well. We have not recovered a snail kite with the vacuolar myelinopathy yet, but it's very difficult to diagnose the lesions because if a bird is not recovered within 24 hours, you start to get autolysis of the brain. And so you can't tell if it's just breaking down in the brain or if they're true lesions. So we can't get a definitive diagnosis. However, the toxin doesn't break down easily. So we can test for toxin in birds of prey that have been exposed to even hot Florida weather for a while. Right. And so we have determined that the fish do develop vacuolar myelinopathy, as well as the snakes, the salamanders. So we can't call it avian vacuolar myelinopathy anymore. Now we just refer to it as vacuolar myelinopathy. But like with most things, probably the biggest thing that we need to manage is not the fish, it's the people, right? People tended to be in favor of hydrilla control once they understood that the hydrilla was killing their fish and wildlife. But this was sort of a novel finding. So it took a while to convince people that those plants look great, and you have lots of birds coming to Thurman now that didn't used to come there, but they're dying here. So that is not a good thing. But we did find at the end of the survey, if you inform them about the potential for wildlife deaths, that we saw a lot more support for hydrilla control. And most of the people would like to see native vegetation only or less hydrilla. Only a few, and they might have been the anglers, wanted the current amount. But, you know, as I said, it's all about education and letting people know what happens what are the consequences of no action. And that is what the Army Corps of Engineers determined, that there were too many bald eagles dying at Lake Thurman, and the lack of action there was being considered a take by the U.S. Fish and Wildlife Service. So they kind of forced their hand to do hydrilla control in Thurman, and they have, and they stocked enough grass carp that we no longer have eagles dying and taking pictures of them lying dead in their nests in the fall. Instead, we're seeing these eagles are actually reproducing in Thurman again. So once they stopped the grass carp and eliminated the hydrilla in Thurman, we have seen the eagles recover. So I think that we don't have to get rid of hydrilla everywhere, and we can probably, but those locations where we have identified that is causing eagle deaths, I think we should get pretty serious about it. And the management solutions are not easy. As this biological control, I think the thing people like to remind me of is, hey, wait, isn't that invasive? So that's not native. Well, it's sterile. The grass carp, really, the triploid grass carp are not able to reproduce. They only live 10 to 12 years, and they do a very good job of eliminating vegetation, maybe more than you want. So there's positive and negatives about biological control, I'm sure y'all are familiar with. And chemical control can be a quick fix for some of these systems and maybe actually help with reducing the amount of grass carp you have to stock if you can reduce the quantity of the invasive plant species before you stock the grass carp. But diquat dibromide specifically contains bromine, and here's my molecule, the Aetokthonotoxin, or toxin that kills the eagle. You see there's 5 bromine atoms on the outside of 2 indole rings. It has an amine group and a nitrile, both of which are supposedly causing most of the toxicity. And it does produce this when there is abundant available bromide in the environment. And I guess y'all know that it's very difficult to harvest hydrilla because it tends to spread it around. And especially if you already had Aetokthonos on it, that might not be the best solution. We tried combinations of putting in carp and some herbicide at Thurman, and followed that up with some sequential stocking so that they didn't have to overstock the grass carp to get the kind of control they needed. And really within 2 years, we had trouble finding hydrilla to collect and to evaluate for autochtonotoxin. So that was beneficial. And we are starting to see more locations across the US where we've documented hydrilla with the cyanobacteria and observed wildlife deaths. All the red watersheds, it has occurred in. Some of these yellow watersheds we've sampled, but we haven't found it there yet. So we continue to receive samples to check for new locations. We did have this, the toxin discovery work published in Science, and since my yes, Timo Niedermeyer did so much of the natural products chemistry in his lab that I would like to show you this so that you can see the part he played in this too. Let's see if it show up.
The AAAS Newcomb Cleveland Prize, our oldest award, recognizes an outstanding original research paper published in Science each year. The winning paper is chosen based on the quality of its scholarship, innovation, presentation, likelihood of influencing its field, and its wider interdisciplinary significance. This year, we present the prize to the paper entitled Hunting the Ego Killer, a cyanobacterial neurotoxin causes vacuolar myelinopathy, first published March 26, 2021. The paper was the product of an international collaboration co-led by Susan Wild, Associate Professor of Aquatic Science at the University of Georgia, and Timo Niedermeyer, Professor of Pharmacognosy at Martin Luther University, Halle-Wittenberg in Germany. At DeGray Lake, Arkansas, in fall 1994, a mass die-off of bald eagles initiated a wildlife investigation. Dozens of birds died, and many displayed odd behavior, like missing their perches and flying into rock walls. When wildlife biologists examined dead eagles, they found extensive lesions throughout their brains and spinal cords. By 1998, scientists knew that birds from at least 10 different southeastern reservoirs in 5 states had this new disease called vacuolar myelinopathy, or VM, but no one knew the cause. In addition to avian species, Susan Wild and UGA colleagues documented that VM could affect amphibians, reptiles, and fish. Researchers made gradual progress uncovering the cause. Wild and her colleagues in South Carolina, where many of the birds suffered from VM, found dense growth of the invasive plant Hydrilla verticillata. The plant, imported from India for use in aquariums, was overrunning man-made lakes in the southeast, readily consumed by coots, which are frequently eaten by bald eagles. Using fluorescence microscopy, Wild observed that in affected water bodies, hydrilla leaves were densely covered with cyanobacteria colonies. This novel cyanobacterium was also found in all the reservoirs where the birds had died. Wild and her colleagues speculated that the cyanobacterium produced a toxin responsible for the eagle deaths. They fed plants collected from affected water bodies to laboratory birds, confirming the plants colonized by cyanobacteria caused VM. The cyanobacterium was named Aetokthonos hydrillicola, Greek for ego killer living on hydrilla. However, no known toxin was detected in the cyanobacterium. In Germany, Niedermeyer and co-authors grew this cyanobacterium in the lab. However, lab-cultured bacteria did not cause VM in test chickens. Hypothesizing that the toxin might be produced in lakes, but not in the lab, they collected more hydrilla leaves from impacted lakes. Direct analysis of the cyanobacteria growing on these leaves revealed a compound not present in lab-grown Aetokthonos that contained 5 bromine atoms. The team was able to reproduce the compound in the lab when the cyanobacteria were grown in the presence of bromide. The compound was revealed to be a novel biindole alkaloid, and further experiments confirmed it causes VM. The scientists called the compound Aetokthonotoxin, toxin that kills the eagle. Jana Marish at the Biology Center of the Czech Academy of Sciences led the genome sequencing of Aetokthonos hydrillicola, which allowed the identification of the compound's biosynthetic gene cluster. Bromide can leach from rocks, but it can also be introduced into the environment from human activities such as coal-fueled power plants or water treatment facilities. The authors recommended increasing monitoring and public awareness campaigns be implemented for hydrilla and Aetokthonotoxin to protect both wildlife and human health. The study brought researchers together from multiple countries and disciplines to solve a decades-old mystery and reminds us how dangerous human-induced changes to ecosystems can be. These unanticipated effects are only likely to increase as we continue to alter the world around us. Hunting the eagle killer, a cyanobacterial neurotoxin, causes vacuolar myelinopathy, recipient of this year's AAAS Newcomb Cleveland Prize. The trip.
No, I don't think we'll watch it again. All right. So in that video, we did run a chicken feeding trial with the cultured cyanobacteria, and we're highly disappointed when we didn't get lesions. We thought we had it, but it turned out that you had to add bromide to the culture media in order for it to produce it in culture. So in the end, we were able to show that bromide availability could make a big difference in the environment too. So more recently, some other folks from Timo's lab have characterized several other toxins that are produced by Aetokthonos hydrillicola. And these dolastatins are actually water soluble and could present, if both are being produced, which it produces these statins all the time, it only produces the toxin when it has the bromide available. So when it's producing both of them, it makes that toxin more dangerous. And we have some new techniques to be able to see it, and you'll hear more about that from Teresa, but Erica Klar in my lab started the project here with the PCR that she got from Linka Stemklova, who's from Czech Republic. I'll show you her work too. But she was able to start screening for this cyanobacteria colony using the red sunglasses and a green light. And this actually is a nice way to be able to see it on the leaf at a larger scale. And I also want to show this map of the locations across the US with the MaxEnt model. If it's in red, that watershed is a very probable habitat for Aetokthonos if you have hydrilla invade there. And we see that throughout Florida, we have those conditions on this ridge that goes down the middle of Florida, all the way down into the Everglades. So even with our earlier model, it suggested that we could be having problems down in Florida. And we did a more extensive survey of hydrilla that was sent to us by a lot of different people from different agencies across the southeastern US. And we found a number of news sites and one very interesting news site that was in New York. And it was an apparently Aetokthonos, but perhaps a different subspecies. But I won't get into that because that's Teresa's story. But we did find a lot of new locations in Louisiana and in East Texas where we had found it before, but there were even more lakes that were affected than we realized. And a new lake up in North Florida. So the toxin characterization, we were able to actually finally characterize it by doing this series of bioassays, starting with our chickens and the cell lines, but we also exposed these invertebrates and larval zebrafish, which make a very good bioassay because zebrafish have a lot of the same genetic material as we do. So the way it affects zebrafish is a pretty good indication of how it will affect the mammals. And we knew that we saw positive results in zebrafish from both Thurman and the Lake Toho down in Florida. But we have some hydrilla control lakes where we never see problems. So we can say it's It's not the hydrilla, it's the cyanobacteria plus the hydrilla. And the zebrafish are tiny. They come from an egg with a chorion to this stage, 7 days old, where they have these little pectoral fins. But if they're exposed to the toxin, they can't right themselves. They barely move around in the petri dish. This one's just upside down. So these are all have been bath exposed to the toxin, and you can see that they are not swimming like the little fast solvent controls anymore. And Tabby produced that work, as well as did a lot of the histology with the fish and with salamanders. So we were able to do the bioassay guided toxin isolation, and that's what really brought us to be able to say this peak right here, that's where that toxin is. If you look at an HPLC trace of all the other things that are left in that extract, that was a very important peak. And so we were able to isolate just that peak and describe it. So that only occurs when in the fall, when bromine is available out in the field. In the summer, we could test it on these bioassays and it was safe. But in a control lake, those extracts were also safe for consumption. This is Timo's slide from the science paper, but it shows you exactly where the cyanobacteria is and the precise signature of that compound, the bio-indole alkaloid with all those bromines. And we found lots of bromine in the hydrilla itself. And even in Lake Okeechobee, which is not a lake where we've ever found the Aetokthonos growing. So it has to be bioavailable too. And there's a trick to that. This is a lower nutrient system, a lower pH, and lower alkalinity and hardness tends to support, that's a VM reservoir that will support the Aetokthonos growth. And it has a lot of bromine in the plants, in the sediment, and some in the water, but mostly it's in the plants. It's much more accumulated. And we knew that once we did the final extract test on the chickens and they demonstrated that they had the same vacuolar myelonopathy, that we could take it all the way back around and blame it on the extract and the cyanobacteria. And even under transmission electron microscopy, we could see the intramyelinatic edema that is the characteristic description of this disease. And we found toxin in wild fish tissues, fish that people consume, you know, especially the largemouth and bluegill and red ear sunfish were pretty high in terms of toxin concentration. Interestingly, the highest concentrations we have ever measured are in the hydrilla. So I keep saying, like, that's the source of this biotoxin. We got to get rid of the hydrilla or we're going to be moving it into the food chain. And as I've mentioned, it's persistent. It's persistent in the hydrilla. It's persistent in the fish and wildlife. And so we were able to demonstrate that that toxin was also in the wild coot tissues that were dying in the field, and the breast tissue and thigh tissue, which can be consumed by hunters, is where it was concentrated. So we know that toxin is neurotoxic, lipophilic, which means it's not water soluble. It's more likely to be trophically transferred, and it can accumulate in wild fish and game. And we are starting to get a lot of very good techniques for detecting it in the field. Genetic detection of the cyanobacteria itself is now quite possible with PCR and with real-time PCR. And we can, we're developed a LAMP assay, which is a little bit easier PCR to be able to do in the field. And we're still working on getting that field application finished. But as far as the laboratory method is complete, and we're able to use these genetic detection techniques to see if that cyanobacteria is present. And the extraction has gotten a lot more streamlined. One of my students, Seth McWhorter, just finished his PhD last year, and using a Quetchers, which is a kit you can purchase, allows you to do quick, easy, cheap, effective, rugged, safe extractions and not use a lot of those dangerous solvents that I was describing earlier. And we have even been able to replicate our results on this laser MALDI-TOF MS sample plate and see the molecule pop up, and the entire Aetokthonotoxin shows up as well. So we have some ways of looking for this toxin. We'd like to get an ELISA method developed so that more folks could test for it, but that's all sort of still in the works. And I'll finish up with just some of the field data that we have from mammals. We did find a sick beaver at Thurman during the time when they were having the outbreaks, and there were dead eagles and coots. And we found a lot of hydrilla in the gut of the beaver, as well as the Aetokthonos hydrillicola in the beaver guts. And it had a very high level of toxin in the gut and in the tissues. And the Southeastern Cooperative Wildlife Disease Study did look at the histology and determined that both the cerebrum and cerebellum had the white matter vacuolization that's common in VM animals. We also saw this type of lesion in Florida panthers. Now, I must preface this by saying many things can cause vacuolar myelinopathy. This is not the only toxin that can cause that, but we did find a toxin in these panthers. So I have concerns that this disease that's affecting the panthers and bobcats and causing hind limb ataxia, and we've already demonstrated that it's in that environment, is going to be more research is needed on this, but I am afraid that this is linked to Aetokthonotoxin as well. And the pythons, to mention them, as long as we're talking about invasive species. So recent research has done that we think a lot of the small mammals in the Everglades have been consumed by the python. Okay, so they're eating all these things, so they have the toxin in them. And we have some locations within Florida where we have demonstrated that there's a toxin, that there's hydrilla present, that there's apple snails, and there is toxin in the python tissues. I'm showing you here in purple some python tail tips that were collected in the Everglades, Francis S. Taylor area. And then we have some panthers and bobcats with the toxin in this area, which is all panther habitat in South Florida. And I think I'm actually about to run out of time, but I will finish up by telling you we would like to get better at telling this story and maybe tell it in a slightly less scary way so that more people can understand and can help us look for locations at risk, and also help us with eliminating hydrilla from sites where we know there's known risk. And this is not finalized data, but these are all the things that we have recently determined Aetokthonotoxin concentration in from the wild. So we have some pretty scary numbers, and the hydrilla Aetokthonotoxin concentration is still the highest. We did complete a mouse trial and demonstrated that all of the mice that were exposed, that were given the extract, the pure toxin, did develop lesions, and we didn't see neuropathy in this trial or anything that we could document it as far as behavior, but they all ended up with lesions, and we found very high concentration of toxin in their bodies at the end of the trial, and quite extensive lesions in the hippocampus, which is basically telling the rest of your brain how to work. We still found some in the sediment, so we're concerned it may remain in the sediment even after hydrilla is gone. So we have a lot of more work to do, but this kind of story keeps expanding, and I would like for more people to be involved with it. And we now have the mode of action, which is published, and we'll learn more about that in the next talk, I'm pretty sure. And I will close now, and I know that we're going to have questions at the end of both talks, so I will look forward to questions.
Thanks, Susan. So we will move to the next talk, which is RT-PCR assay development of ITS and toxin gene regions of Aetokthonos hydrillicola, a known epiphyte of the invasive species Hydrilla verticillata. And Dr. Teresa Hattenrath from New York State Department of Health, Wadsworth Environmental Biology Lab will be giving that talk. So Teresa has 19 years of experience in harmful algal bloom ecology. She earned her MS and PhD in marine sciences from the School of Marine and Atmospheric Sciences at Stony Brook University. Her graduate research focused on assessing the effects of nutrients and ocean acidification on the growth and toxicity of harmful marine dinoflagellates. In 2017, Teresa joined New York State's Department of Health's Wadsworth Center as a research scientist in the Laboratory of Environmental Biology, where her primary focus is on freshwater harmful algal blooms. She currently serves as the laboratory chief of the Laboratory of Environmental Biology. Take it away, Teresa.
Hi, everyone. I'm really excited to present this collaborative project between my group at the Wadsworth Center at the New York State Department of Health and Dr. Kathy McGlynn's group at the New York State DEC Invasive Species Unit, as well as Dr. Susan Wild's group at the University of Georgia. Oh, my slides are not advancing. Hold on here. Let me restart that. Oh, there we go. Okay, so Dr. Wild gave a great overview of this ecosystem risk that's associated with the invasive aquatic vegetation. So I'm just going to briefly summarize. So the vacuolar myelinopathy was first discovered in bald eagles and waterfowl in the southeastern United States back in 1994. These large-scale die-offs led Dr. Wild's group to focus investigations on submerged aquatic vegetation, which ended up leading to the discovery of the epiphytic cyanobacteria, Aetokthonos hydrillicola, and that was again found on the invasive hydrilla. So by feeding ducks these hydrilla infested with the Aetokthonos, Dr. Wild's group was able to determine that Aetokthonos was what caused vacuolar myelinopathy. However, it wasn't until 2021 when the structure, toxicity, and biosynthetic gene cluster of Aetokthonos hydrillicola, the neurotoxin that's produced by that organism, which is called Aetokthonotoxin, was fully elucidated. And so through additional studies by Schwark et al., they discovered that the pure Aetokthonotoxin was actually less cytotoxic compared with crude extracts of A. hydrillicola from culture. And then this ended up leading to the discovery of a second toxin that was produced by A. hydrillicola called Aetokthonostatin. So like all this interesting research got my wheels turning because I knew that the invasive hydrilla was present in New York State. So it was first discovered in New York State and Orange County in 2008. And it has been found in other New York State counties, which you could see here from this distribution in red. And there's actually a well-established population in the Croton River. And so as a harmful algal bloom ecologist and someone who works at the Department of Health, I really thought that we needed to get ahead of this in New York State and monitor for this because it is a potential human health problem. As Susan said, it can propagate up the toxin can propagate up the food chain. So I ended up contacting Kathy McGlynn at the New York State Invasive Species Unit, and found out that in 2023, the Wild Lab had already confirmed the presence of Aetokthonos hydrillicola that was associated with the hydrilla in Lake Sebago in New York State. So I told Kathy that I was confident that my lab group would be able to create a real-time PCR assay that we would be able to use as a monitoring tool, and an awesome collaboration was born. So our group's study aims were to develop a fast and accurate method for detecting Aetokthonos and its toxins in aquatic plant materials by targeting the ribosomal RNA ITS region and the biosynthetic gene clusters of Aetokthonotoxin and Aetokthonostatin. And we ended up targeting two sets of primers and probes within those gene regions, so that way we could see if there was any differences in those. The next thing we did was we wanted to conduct a survey of New York State lakes to determine the occurrence and distribution of Aetokthonos and its toxins. So for this presentation, I'm going to briefly cover the RT-PCR assay development, including the assay optimization and validation, the specificity testing that we did, the blinded sample study we did with the Wild Lab, and then I'm going to jump into the New York State surveys that we conducted in 2024 and 2025. And then I'll briefly talk about our efforts to sequence the northeastern Aetokthonos hydrillicola variant. So for our RT-PCR assay, the primers and probes and a synthetic DNA standard that we used were all developed using sequences that were available on the National Center for Biotechnology and Information Database. It's also called NCBI. Our gene targets included the ribosomal RNA ITS region, the Aetokthonostatin region, and the Aetokthonotoxin region. And those were all the regions from sequences that we found on NCBI for Aetokthonos. So we ended up choosing these primer probe pair sets based on the product that they produced. So you can see here that all these products are less than 150 base pairs in length, which is actually the sweet spot for real-time PCR. And before we ended up conducting our assay validation, we conducted several comparison studies so that way we could optimize our reverse and forward primer concentrations and probe concentrations. And these are the final concentrations that we landed on for those assays. So I just wanted to talk briefly about the synthetic DNA standard that we created, which it's called a G-block. It's a really cool piece of technology that you could get synthesized for you by the Integrated DNA Technologies Incorporated, also called IDT. It's actually something I learned about when I was a graduate student attending a conference, and I've used this cool piece of technology in my research ever since. So if you're a lab person like me, I really highly suggest using this because it's actually more streamlined compared to using things like plasmids as standards, because this already comes as a linearized piece of DNA. So just to kind of briefly show you, so what happens here is you end up linking all of your target sequences together. So they're called amplicons. So you'll have a flank piece of DNA, your amplicon, a spacer, another amplicon, and so on and so forth. So this is what our synthetic piece of DNA looks like. We have the flank regions here and here. In the red, we have all of our spacer sequences. And in between, we have all of our full-length target sequences for the ITS, our RNA primers, the Aetokthonotoxin, and the Aetokthonostatin. So just kind of show you an example here. So here's one spacer, then here's the forward primer, then there's the probe, and then the reverse primer for that sequence. So this G-block, we ended up using for assay optimization and validation. And we also used this as a positive assay, one of our positive assay controls. So we validated these assays over a 3-day time period, and we did this by using 10-fold serial dilutions of our synthetic DNA standard, starting from 125 million copies per reaction, all the way down to 12.5 copies per reaction. So we plated this and then ran this on our QuantStudio 5 RT-PCR. And so here you can see our results. I'll just walk you through this. So this is the mean CT values and the log copies per reaction. And these are the standard curves for all of our targets. So again, for the ITS regions, the atokthonotoxin regions, and the atokthonostatin regions for both singleplex and duplex assays. So singleplex means that you'll only be looking at one of these targets, and then duplex means that you'll be looking at 2 targets at the same time. So that basically saves reagents and such. So standard curves were actually within ideal parameters that you would want for a typical RT-PCR assay. So we found slopes between -3.6 and -3.1, which also corresponds to a 90 to 110% efficiency. And then for the coefficient of determination, or the R-squared, all of these assays were more than 0.98. We also found that all of our singleplex and duplex assays were linear between 12.5 and 125 million copies. And all of our singleplex and duplex assays, as you can see, they're kind of just laying right on top of each other there, had similar R-squared values and amplification efficiencies, which is great because that means that you don't get competition between 2 duplexing targets. For these assays, we found that the limits of detection were specific for each assay and were either 1.25 or 12.5 copies per reaction. So the next step for creating this assay was to determine whether or not the primers and the probes were specific to the target genes that we were looking at. So we determined this by testing 40 phytoplankton cultures of co-occurring species of Aetokthonos hydrillicola. So of course, Aetokthonos hydrillicola here represented our positive. So it should have the ITS region and the atokthonotoxin and atokthonostatin regions present. But then we also tested what we assumed would be negative cultures, so Selenastrum, Synechococcus, Lyngbia, Cylindrospermopsin, Oscillatoria, Chlamydomonas, Aphanomizenin, Anabana, Dolichospermin, and Microcystis. So what we did is we ended up growing out these phytoplankton cultures. We lysed these cells, then we put these lysates into a plate compared to our synthetic DNA standard curve, and ran this on the RT-PCR for all of our gene targets. And what we found, fortunately, is that the only thing that came up positive was Aetokthonos, as we had expected. So the next step here was we wanted to determine if these newly developed RT-PCR assays correctly detected all the target genes, but if they did so in field samples, because that's ultimately what we want to look at. So in order to do this, we turned to Dr. Wild's group at the University of Georgia. So they actually had a whole bunch of samples that they had already extracted, and they ran on their LAMP assay, also known as the loop-mediated isothermal amplification assay that Erica Klar was working on, and then the traditional PCR assay that Lenka was working on. We took these samples, they were sent to us in a blinded fashion, and we analyzed them using our newly developed RT-PCR assays. So what does blinded mean? So that basically means that they sent these samples to us without us knowing what the results were or where these samples were from. So we took our results and we sent it to Dr. Wild's group, and they confirmed whether or not the assay worked. So here's a table of those results. Here you could see that they sent us an extraction positive control, which was their Aetokthonos culture. And they sent us 5 samples that were collected from lakes around Louisiana, Texas, and North Carolina. So again, we ran our RT-PCR assay data, excuse me, our RT-PCR assay, and this targeted the ITS region, which specifically looks for the presence of hydrillicola. It targeted the Aetokthonos toxin and the Aetokthonostatin gene regions. And again, our comparator assays were the PCR and LAMP assays at Dr. Wild's lab that looked for the presence of hydrillicola and Aetokthonos toxin. At this time, there were no comparator assays for Aetokthonostatin. So what you can see here is that our RT-PCR assay results compared to the comparator assays all matched up, which is great because this means that our field samples, our assay was almost ready for analyzing field samples. So before we analyzed field samples, we also wanted to test our extractions in field samples. So we decided to evaluate different extraction methods. We already had an in-house extraction method that we used for fecal indicator bacteria via EPA 1609. This extraction method made use of a tube with glass beads in there. And then we compared that to Dr. Wild's extraction method using a D&EZ plant pro kit, which makes use of this tube with a single metal bead. And just kind of an example to show you what we found in the initial tests, if you look over here, look at the mean CT values and all of our targets here, you could see that the in-house assay, the in-house extraction method, which is in white, had a lower CT value than the D&EZ assay, which is in gray here. And that happened for all of our targets. So what we wanted to do is we wanted to go back and we wanted to add another modification. So we ended up testing the D&EZ Plant Pro Kit using the same bead beading tube that we use for EPA 1609. And so we then ran all of the samples using all of these different extraction methods. And we basically found that when you run the D&EZ Plant Pro Kit using the EPA extraction method tubes, you actually get very similar CT values. And so ultimately we decided that smaller glass beads are more efficient at extracting the epiphytic cyanobacteria from aquatic plant material. So now we were ready to analyze field samples. So if anyone is interested in learning any of the specifics, we actually recently just published this RT-PCR assay method in the January 2026 issue of Harmful Algae. All right, so now I'm going to move on to talk about my New York State surveys. So we wanted to see, again, if Aetokthonos hydrillicola was present in New York State water bodies. So to do that, we conducted surveys across New York State, and we looked for hydrilla and Eurasian water milfoil samples, which were collected using rake tosses. So these surveys were actually a huge multi-agency effort, and we have a lot of people to thank for these samples. So these samples were sent to my lab, And as Dr. Wild was showing you in some of her slides, we used a similar setup where we used a night sea fluorescent flashlight that is green, and we used red glasses so that way we can look at the red fluorescent cyanobacteria on the leaves of the aquatic plant material. So we took 80 milligrams of sample from all the samples that we got, we extracted them, and then we prepared that lysate compared to standard curves and ran that for all of our newly developed assays on the RT-PCR. So in 2024, we basically focused on sampling Rockland County, so that is where Lake Sebago is located. That's where Dr. Wild's group first found Aetokthonos in 2023. We also focused on several areas in Suffolk County, which had unmanaged populations of hydrilla and Eurasian water milfoil. So here are the results that we found from that 2024 survey. So each of these water bodies had several sites in which we sampled hydrilla and Eurasian water milfoil. You could see here that Suffolk County sites had were all negative for ITS, atokthonotoxin, and atokthonostatin. And this indicates that Aetokthonos was not present at any of these sites. But if you look here, Lake Sebago in Rockland County, that actually tested positive for atokthonostatin genes, but were negative for both the ITS regions and the atokthonotoxin regions, indicating that Aetokthonos is potentially present, but I'll talk a little bit more about that later. So in 2025, we expanded our surveys. So actually, once again, we sampled in Lake Sebago since that came up positive for atokthonostatin. We then expanded out to Westchester County and the Croton River and the New Croton Reservoir. We went to St. Lawrence County, Hickory Lake, Owego Creek and Spencer Pond in Tioga County, Cayuga Lake, which actually spans Seneca, Tompkins, and Cayuga counties, Tinker Nature Center Pond, which is located in Monroe County, Little Niagara River, which is located in Niagara County, and finally we went to Erie Canal and Green Lake in Erie County. And what we found is that the hydrilla and Eurasian water milfoil samples from all of those surveys were negative for ITS, atokthonotoxin, and atokthonostatin genes, indicating that Aetokthonos is not present in any of those samples we tested, thankfully. So this actually includes Lake Sebago hydrilla samples, which were collected from 3 different sites within that lake. And remember, this is the location where in 2023, Dr. Wild's lab found Aetokthonos was present. In 2024, we found that it was positive for atokthonostatin genes. And as it turns out, this particular site is managed with fluoridone treatments, which are causing chlorosis and leaf defoliation, which you could see in this picture right here. The hydrilla is looking very unhealthy. The leaves are turning clear. And this also means that the hydrilla is unable to support that Aetokthonos epiphyte. So this is actually a really nice example of management in action. So in 2026, we plan to conduct additional surveys. We plan to test the same sites. In addition, we're going to add Niagara River as a sampling site. And then if any other sites come up as hydrilla positive, we'll test those sites as well. All right, so now I'm just going to move on to talk about the sequencing of the Northeast Aetokthonos hydrillicola variant. So recall in 2024, I had mentioned that Lake Sebago hydrilla samples tested positive for the autochthonostatin genes, but they were actually negative for both the ITS region and the autochthonotoxin genes. You might say to yourself, well, how are they positive for the autochthonostatin genes when the organism Aetokthonos, which is, you can test for that, the presence of that gene using the ITS region, is negative? Well, that doesn't make any sense. Well, I was able to confirm the presence of Aetokthonos hydrillicola-like cyanobacteria, as you could see from this micrograph, on the underside of the leaf of hydrilla from Lake Sebago. So you could see here the dense branching dark filamentous colonies that are typical of Aetokthonos hydrillicola. And so what we think is happening here is the difference between the results that we're getting, where, again, I said that the gene regions were coming up negative for ITS, which is what actually means that Aetokthonos is present. But in microscopy, we're actually seeing that a hydrillicola-like organism is present. So what I think is happening here is that Lake Sebago actually hosts a Northeast genetic variant, and that's different from those in the southeastern United States. So remember, I used those southeastern US sequences in the NCBI database to actually create my primers and probes for this assay because there were no other sequences available for those. So what we wanted to do is we wanted to determine if this was indeed a different genetic variant. So we sequenced the Lake Sebago ITS region, so that way we can compare that back to the sequences from southeastern United States that are already in the NCBI database. So we took high fidelity GOTAC master mix, mixed with primers that were in the Wild et al. 2014 paper that were specific to the ITS region. We conducted PCR. We then PCR purified those products. We then took those ITS region products from Lake Sebago and we inserted that into a plasmid. That plasmid was then transferred into a E. coli cell. So basically what's happening here is this E. coli is doing the work for us and basically replicating all of these sequences. So we grow this out further. After it's done growing out, we end up purifying all those plasmids, and then we sent those plasmids in to get sequenced at our genomics core at the Wadsworth Center. So here we have Lake Sebago sequences aligned with the sequences that were available in NCBI. So that's all those southeastern United States sequences. You could see Florida, Thurmond, Florida, Thurmond, all here. And you can see that by the percent identity, that our Lake Sebago variant is only 97.74% at the highest identical to other Aetokthonos hydrillic sequences in the NCBI database. So what my lab is now doing is we're resequencing gel purified amplicons to get better sequences, so that way we could actually create primers for this Northeast variant. I kind of wanted to end this on a little warning, I guess, a word to the wise. So for anyone who wants to incorporate this method into their monitoring program, I highly suggest that you're aware that not all tissue lysers are created equal. So I will say this is not a knock on either of these companies. We actually use both these instruments in our lab. but we've done rigorous testing to ensure that we're getting the same results. Because as it turns out, bead beater speeds are not the same across all tissue lysers, so you might have to tweak the time in order to get it to produce the same results as the other. Okay, so in conclusion, a fast and accurate method for detecting Aetokthonos and its toxins in aquatic plant materials was developed and will allow for rapid screening of lakes. Our data suggests that Aetokthonostatin genes are likely conserved across genetic variants of Aetokthonos and might actually be the best marker for screening for the presence of Aetokthonos hydrillicola in lakes. Again, my lab is currently resequencing the ITS region of the Aetokthonos from Lake Sebago, so that way we could create primers and probes for this specific genetic variant. And word to the wise, don't assume that tissue lysers produce the same results. And with that, I'd like to thank a whole bunch of folks from my lab here, from Dr. Wild's lab, from Dr. Kathy McGlynn's group at the Aquatic Invasive Species Unit. And again, like I mentioned, we have so many people to thank for collecting samples. That includes all these folks from the DEC, Little Bear Environmental, the New York City Department of Environmental Protection, New York State Parks, and then the invasive species management groups from both the Finger Lakes and Long Island. And with that, I will stop here and take any questions.
Okay. So please feel free to put your questions in the chat. And while people are writing, I have a few, so I'll go ahead and ask. So some questions for Susan. Do you foresee in the near future having more people investing in research on additional vertebrates?
Yes, because Sonia Hernandez is going to be taking over the research with Tobias Hames, and she is a veterinarian as well as a wildlife biologist and intends to continue working on this. She works for Warnell as well as SWIS, so she has two hats, and she makes a very good person to continue this research.
Okay. And as far as the field test goes, where is that? You had mentioned that during your slideshow. We were talking about some sort of rapid testing that could be done in the field.
Well, we would like to have an ELISA for the toxin, right? The same way we do for microcystin and some of the other cyanobacterial toxins. And the reason that we couldn't do it directly is that Aetokthonotoxin is actually a pretty small molecule, but Timo has recently conjugated it, which means you can attach some colorimetric responses to it. So we're making some progress on that, but I think Teresa should address the LAMP, which is something that you can detect the genetic material and determine that that cyanobacteria is present. And I especially like that she mentions the Aetokthonostatin, because we saw that with ours, it was always produced Aetokthonostatin, so it's a good indicator molecule.
Okay. And final question regarding Florida. Is there a lot of education and outreach or discussion about addressing these issues regarding the presence of Aetokthonos? Because I know that Florida has a pretty extensive hydrilla infestation that they're managing throughout the state.
Yeah, and the locations where we've already identified that Aetokthonos hydrillicola is present that is in some of the managed lakes. So there are times when they don't collect as much hydrilla there because they are controlling it. But I do have concerns about that interaction with the herbicide causing a dieback of the hydrilla, which might promote the cyanobacteria in a lakes in Florida, which wouldn't normally the hydrilla wouldn't die back, right? So I think there's a concern with understanding that management is really important, but you have to use something that's, well, could be more long lasting. And I guess that was what happened with Lake Sebago.
Okay.
Fluoridone is more expensive, but it does seem to do a better job at a longer control. much more effective.
So we have a question in the chat. Have there been any documented findings of hydrillicola on Eurasian water milfoil or other aquatic plant species besides hydrilla?
Yes, we did find it on Eurasian water milfoil, Egeria densa, and several of the native plant species.
Okay. And while folks are writing their questions in, I have some questions for Teresa. So you were
Sure.
looking at the, I was wondering if you're going to be doing additional research on subspecies and how the variants differ from one another genetically and through the production of the different molecules.
Yeah, that's a good question. Well, like right now, we're just resequencing the ITS region so we can really see what regions are different, because if you recall from the slide, there's a 97.74% difference between the northeastern variant and then the variant that is in the southeastern United States region. And we found that while it is the gene for atokthonostatin is present, it doesn't look like the gene for atokthonotoxin is present, which is interesting because that's very different from what Dr. Wild is seeing in the southeastern United States. And you know, this has happened before in other toxic species where you'll see that genes have been deleted over the evolutionary period. So it might be that that's the case, but we just don't know yet.
Okay. Okay.
It's interesting to think about the origin, right, of
Yeah.
the Aetokthonos, because we don't yet know. Did it come in with dioecious or monoecious hydrilla as a co-invasion, or was it already here, but just in low abundance until it had the perfect substrate to just explode in our lakes and grow on? And we really don't know yet, but I think having a clue like a non-toxic version is really helpful, because did it lose its ability to make the toxin, or did the southeastern version
Gain it. Yeah.
gain it?
Yeah, exactly.
So do we have any, again, I don't know, I don't have that in-depth knowledge of research outside of the US, but do we have any sense of in the countries where hydrilla has come from originally, has there been any research regarding the presence of this type of cyanobacteria?
No, I had really always thought I'd be able to get over to China or look at some samples from the, countries of origin, but I really haven't followed up on that. That's something I think someone else should be working on. It's curious that in its native range, the monoecious hydrilla is wimpy. Like they don't have any trouble with controlling it.
Mhm.
So what has it done here in the US that's making it so terribly invasive? It's different.
Yeah.
I think it's definitely that's one that should be tracked back. Teresa, you need to work on that.
Yeah, I mean, it's interesting too because, yeah, I've been actually looking in the literature to see if there's any Aetokthonos in, Europe and Asia and whatnot, and they're not, well, it doesn't look like they're seeing it there. There's no reports on it, so it's really interesting because does that mean that the Aetokthonos was here in the US first, and then it just started growing on that, the hydrilla because it had the perfect substrate. So we just don't know.
Okay. That's fascinating. Yeah. Any other questions folks want to put in the chat box?
Well, I appreciate people's interest in staying on the webinar for so long, and welcome questions that you might come up with later if you want to send those. Our contact information will be available.
Yep.
That'd be great. Well, I want to say thank you to both of you. It's been a privilege.
Thank you.
Thank you.
And I do want to, let me just share my screen for a moment. I just want to do my little pitch for the next session. So let me do that. So stay tuned, folks, if you're interested. There's a session 3 in this webinar series, which will focus on European frogbed and Phragmites. And again, I'd like to thank both Susan and Teresa for taking the time to give presentations today and just in general supporting the work that's being done by lots of members of the Northeast Aquatic Plant Management Society. And I hope that we get to continue working together for quite a while. Thank you, everyone.
Thank you.
Thank you.
Have a wonderful afternoon. Bye.
You too. Bye.
Bye.